Hello and welcome to chapter 22. We are going to talk about reproductive physiology. We're going to talk about gamete formation. But before we jump too far into that, I think it would be important or maybe even behoove us to really talk about what meiosis is. We focus a lot on mitosis in AMP1, we focus a little bit on it in AMP2, and then when you get to patho, we're going to talk about it in regards to cancer. But meiosis is just kind of skipped over, and it's really, really important. It really drives the entire chapter here, and it drives our species' entire ability to procreate and perpetuate the species. So meiosis is considered a reduction division where mitosis is just clonal expansion. So one cell basically creating two identical copies of itself. Where meiosis, on the other hand, is not. Meiosis, we have to start thinking about two new terms, one being diploid and one being haploid. So what that means is you are a diploid creature. A way to abbreviate that is the number 2 and then the capital letter N, 2N. And that means that you have 23 chromosomes that are duplicated. So there are copies, but they don't have to be identical copies. You could have different forms of genetic information on each chromosome, but it just means you have a pair of each of the 23 chromosomes, giving you a total of 46. The reason this is so important is because our sex cells, the X and the Y chromosome, they're unique. They are dividing out genetic information to unite and form a zygote, which will eventually be the next member of our population, a baby. So male sex cells are sperm and female sex cells are called eggs or oocytes. We're going to go into all the details of how they're created and maintained throughout this chapter. So meiosis is 2 cell divisions. Remember, let's think back. Mitosis is 1 cell division. So meiosis 1 and meiosis 2 each do different things. And there's more vocab, so we're going to kind of go over some of that here. So before meiosis even starts, each chromosome is going to replicate. And you can see this extra picture I've added to your slide. This is not something from your textbook, but it's a really good hallmark representative of a duplicated chromosome. The duplicated chromosome has these arms called chromatids. And you can see I have double arrows here pointing to the chromatids. They're 2 identical chromatids. They are exact copies of one another. And this whole structure is held together at the center by something called a centromere. It's like a little button or a little protein knob that holds everything together. All right, now let's really get into the nitty-gritty of what meiosis is. I told you before that there's 2 cell divisions. There's meiosis 1 and meiosis 2. And I kind of combined 2 slides here because I think that it's really important to follow the picture while we go through the explanation. Where we start here, this diploid cell, this is how a progenitor, a precursor, the original sex cell is going to start. Then we're going to undergo the first meiotic divisions. This is M1 essentially. And what happens during meiosis 1 is you're having those duplicated chromosomes. You can tell they're duplicated because they're the letter X, just like you saw on the previous slide. And you can see those little round buttons, the centromeres, holding everything together. All right, remember, they don't have to be identical. Hang on, let me get my mouse. There it is. This X may not be identical to this X, but they are still representative of the same chromosome. But the arms, they will be identical. So during the first meiotic division, it's similar in what you've learned in mitosis in that we have our centromeres that migrate to the poles. We have our chromosomes line up at the center. We pull them apart. We have our kinetochore. And then what happens is we are pulling apart those paired homologous chromosomes. So we're pulling apart we're going from 46 here to 23 in the next round, okay? When you are down from diploid to haploid, you essentially become haploid here, and then you stay haploid at the end. I'll get to that in a minute, so don't let this confuse you. I should have put the word haploid beneath this. So if you are annotating your own notes, go ahead and add the word haploid, because this is where the first haploid cell is going to begin. All right, so we've gone from diploid to haploid with meiosis 1. We've essentially reduced the genetic information by half. Then we undergo another round of meiosis. And in that other round of meiosis, notice that there's no duplication. There's no X's. So the X here is not going to all of a sudden replicate and get another second X that you see here. What happens in the second round of meiosis is the X's are split at that centromere, at that button that I mentioned. So you're pulling apart the individual chromatids. So then at the very end, you end up with 4 cells that have 23 chromosomes that are haploid that are ready to undergo further differentiation to become sperm or eggs. All right, so let's go through it in more detail. So there are 4 phases in meiosis 1, and I'll spoil the punchline for you, there are 4 phases in meiosis 2. So in prophase 1, you're going to see something called synapsis, and I have a picture of this I'm going to show you in a minute. Synapsis is when you see the pairing up of the X's. Actually, we'll just jump ahead. So here's what we're talking about. This is synapsis. So this chromosome and this chromosome are similar. The genetic information may not be identical, but the genes are going to code for the same things. Like, for example, this chromosome might have a gene for eye color representing brown eyes, and this chromosome will have the same gene for eye color, but it doesn't have to code for brown eyes. It could code for blue eyes. All right, I'm going to go back. So also during prophase 1, the crossover is going to occur. So as you saw on that slide, and I'll go back to it in a moment, you can see that there are places where the sister chromatids are going to kind of intertwine and touch. And when they do that, they can actually flip-flop pieces back and forth. And this is really important because the more flip-flopping of pieces that they do, the more genetic variability that we're going to actually confer into our sperm or our egg. It's called recombination, swapping of genes back and forth. All right, so after prophase, we move into metaphase. And metaphase 1 is similar to metaphase you've already learned about in mitosis, in that all the chromosomes are going to line up at the midplate. And then after metaphase, we go into anaphase 1. So in anaphase 1 is where we start to see those X's being pulled apart. So this is where we start to go from diploid to haploid. And then meiosis 1 is wrapped up in telophase 1, where the cell division is absolutely complete. The 2 new daughter cells are formed, and the nuclear membranes reform around the material, as you saw on the slide before this one. All right, so we've already talked about this slide, so now I'm going to jump into the second meiotic division. So the second meiotic division, there's a really important note that you have to make, is there's no duplication of genetic material. We always talk about mitosis is that before the cell undergoes replication, all the genetic information is duplicated. And we saw that to be true during meiosis 1. So that's how we got our X's. Well, now when we're going into meiosis 2, there is no duplication, none, because we want to pull apart those sister chromatids. So in prophase 2, the chromosomes basically, your book says they condense. What that means is they go from being a big jumbled mess in the nucleus to looking like perfectly linear, straight pieces of DNA. During metaphase 2, we are going to see those X's line up at the midplate. During anaphase 2, we see the centromeres, those little center buttons, break, and the chromatids, so the X's are now being ripped apart, and they go to the opposite ends. And in telophase 2, each of the 2 cells that were produced in meiosis 1 is going to give rise to its own set of daughter cells, so now we're going to go to 4. So we went from 1 diploid cell to 4 haploid cells. All right, So this is just another kind of brief talk about what crossing over is and why it's so important. So this is, again, 2 chromosomes. This one could have a gene coding, let's say, for hair color. This one could code for red hair, and this one could code for brown hair. But they're the same chromosome, just the genes code for different material. Well, as they line up, they might swap something back and forth. So the reason it's so important is because the chromosome doesn't code for just one gene. Even though it was coding for hair color, it could also code for eye color, it could code for height, it could code for any characteristic that you have. And so if we go with traditional division, if this chromosome coded for somebody with red hair and brown eyes that was going to be 5'5", and this chromosome was going to code for somebody with, I don't know, what did I say before? Brown hair with blue eyes that would be 5'2". So you get one person or the other. But now if we start to swap pieces around, we're now going to get a lot more genetic variability, and we're going to instill this, it's actually a strength at the level of the nucleus for propagation of the species. So I know that's seemingly complicated, but the greater genetic variability you maintain in your species, the stronger that species really is evolutionarily. So this is a really important phenomenon. So that was just a really brief overview of meiosis. If you are uncomfortable or unfamiliar with meiosis, I'll post some other really good videos that I like up in the module that will help explain what meiosis is as well. But now we're going to kind of take a sidestep, not in total different direction, but we're going to talk about formation of gametes for male or females. So we're going to talk about spermatogenesis and spermiogenesis, and then we're going to talk about oogenesis for women. So in doing that, we're also going to cover all of the male reproductive physiology, and we'll cover a little bit of anatomy, but you guys should already be abundantly familiar with the anatomy from Anatomy and Physiology 1. So we'll jump in and talk about the functions of the male reproductive organs. So one of the main functions is going to be to maintain the sperm. So to make sure the sperm is essentially born, for lack of a better term, that it's healthy, that it can mature, and that it can leave the body appropriately. Another function of male reproductive organs is transportation of sperm and fluids outside of the body. We will talk about how the renal system the urinary system as well as the reproductive system merge in the vas deferens and the urethra in the male penis. And then we will also talk about the function of the male reproductive system being in hormone production. And hormone production, yes, is important for sex cell formation, but hormone production also maintains a lot of other really important factors in the male body, like bone integrity, and it changes hair patterns, it can affect height, and it can also affect, there is some research that suggests it can even affect temperament. So the primary sex organs in male or female are called gonads. Now, the gonads in a male are 2 testes, so it's a paired structure. And in the testes, the sperm cells or they're called spermatozoa, are formed. And male sex hormones like testosterone are also produced there. There are some other accessory sex organs, internal and external, and we'll talk about those as we go through the lecture. So one important note to make is there is a special type of cell called a Sertoli or a sustentacular cell. I typically call them Sertoli cells. That's a little bit older name. That's how I learned it, but they are the same thing. So these Sertoli cells are really, really important. They're very big cells, and they basically span the entire thickness of the outer layer of skin, the epithelium, not the skin, but the outer coating of the seminiferous tubule. And then the seminiferous tubules is where you get nourishment and supportive chemicals that are secreted to help sperm during maturation. So sperm are essentially born, for lack of a better term, through a very, very specific sequence. So spermatogonia are like the rudimentary precursors. They divide and become primary spermatocytes. The primary spermatocytes become secondary spermatocytes, And as they further mature, the secondary spermatocytes become spermatids. And then when the spermatids are finally able to propel themselves or fully motile on their own, then they become spermatozoa. So there's two terms that you definitely need to know the difference between: spermiogenesis and spermatogenesis. So spermiogenesis is the development of spermatids into fully mature sperm, and spermatogenesis is the combined process of meiosis and spermiogenesis. So spermatogenesis is basically the all-encompassing big umbrella of everything. All right, so let's talk about the formation of sperm cells. So here you start with your primary spermatocyte. All right, we got 46 chromosomes, so this is diploid still. You don't want a diploid cell to go for reproduction. You undergo your first meiotic division, so now we're becoming haploid. So the reason you know we're haploid is because now we're at 23 chromosomes. 23 chromosomes automatically means haploid. You undergo your second round of meiotic division, and you're still haploid, you're just pulling apart those X's, and what you're left with are the spermatids. Now, the spermatids have to undergo lots and lots of maturation, and then they become fully functional sperm cells. So I want to make a note about what this actually looks like. So we see this picture back here, and it's just this really pretty diagram, but what's it actually look like as it's happening in the testicle? This. So here, let's go through this again. So here we have your rudimentary precursors right here, and this, you could put an arrow on your slide right here. This is where we really pick up our story. This is your primary spermatocyte right here. The primary spermatocyte is then going to divide and become the secondary spermatocyte, and then eventually we're going to get to our spermatids. Remember, 1 becomes 4. And then notice this here is the basement membrane. This is essentially the outer area of the testicle, and we're moving more and more and more and more closer interior to the lumen. So eventually we are going to get these sperm that are going to travel in the epididymis. You can see where we're at here in the epididymis, and they're going to eventually travel up and around and propel through the male reproductive tract. But we'll talk more details in a few moments. So we push again from the basement membrane, the outer layer of that epithelium, all the way to the luminal junction. And notice here that the difference between this and this, their sperm are still not mature, even though they're docked right at the lumen. So I should take that back. They might be mature, but they're not fully functional until they are actually propelling through the epididymis. So another view of this. So here again, we have your rudimentary precursors. We undergo all of our divisions, and we undergo maturation steps. Notice we start out with this cell that looks a lot like an egg, and then we basically end up with something that kind of looks like a tadpole. And how do we go about doing that? Well, the maturation process is basically crafting what this entire structure is going to look like. Excess cytoplasm is pinched off and blebbed away, and parts of the sperm are compartmentalized for their function. So there's a head, there's a midpiece, and there's a tail. And the tail can be varying in length. That's what this cross right here means. But what's interesting is this midpiece is where all of the mitochondria are contained. The head contains all of the genetic information, and then a little pocket of enzymes called the acrosome, and they're going to be really responsible for undergoing a process called the acrosome reaction that I'll talk about later. So eventually, when sperm are going to leave the body, they leave in a fluid called the semen. So the semen is made up of sperm plus a whole bunch of secretions that we're going to talk about right now. So the secretions come from the seminal vesicles, the prostate gland, and the bulbourethral gland. So the bulbourethral gland is also called the Cowper's gland. So if you've heard of that, either one is correct to say. Neither one would be wrong. So semen, interestingly, is slightly alkaline. It's got a pH of about 7.5. This is really, really important because the interior compartment of the vagina is slightly acidic. And if the semen was not slightly alkaline and the sperm were to enter an acidic vagina, then the acid could actually potentially destroy the sperm, and then the sperm would never even make it to the egg for fertilization. So the fact that it comes in slightly alkaline is a protective mechanism. There's also prostaglandins, which are really important for actually irritation. And the irritation is important to help propel the sperm to their end goal being the egg. And then the semen is full of nutrients, lots of vitamins and minerals, and specifically a sugar. And it is full of fructose. The sperm are going to survive off of fructose while they are traveling through the female reproductive tract to reach the egg. So there's about between 2 and 5 milliliters of semen created every time a man ejaculates. And interestingly, in that 2 to 5 milliliters, there's on average about 120 million sperm. A man is considered to be not producing enough sperm if he produces less than 100 million sperm per milliliter of his semen. So we feel like these are really large numbers, but they're actually really important to ensure success of sperm to reach the end destination being the egg. So the process that happens during sexual arousal and the physiological deposition of sperm into the female reproductive tract is actually pretty fascinating. So erection is stimulated by the parasympathetic nerve. So we think about fight or flight and then rest and digest, right? Fight or flight is sympathetics and rest and digest is parasympathetics. You might think that there's a lot of activation that's occurring during sexual stimulation and that it would be your sympathetics, but that's incorrect. So it's parasympathetic innervation that drives sexual arousal. And interestingly, the parasympathetic nerve is going to drive the release of a substance called nitric oxide. Nitric oxide is a really, really potent vasodilator. So it's going to take all of the arteries in the penis and cause them to get very, very wide open so a whole bunch of blood can flow in at once. This is called engorgement. And this is when the penis would become hard during the erection phase. So at the culmination of the sexual experience, there is an orgasm. And during that orgasm, it can be divided into 2 subcategories, both emission and ejaculation. Now, interestingly, people often think of this as one, or they get them confused. But emission is going to come before ejaculation. Emission is simply the movement of semen into the urethra. So you're basically moving from the testicles through the vas deferens, up and around the bladder, passing by all of the glands that we talked about, the seminal vesicle, the Cowper's gland, and the prostate, not in that order, and picking up all of its appropriate secretions, and then finally merging with the urethra to exit out of the body. That is the emission phase. Ejaculation begins when the semen is leaving the body, leaving the urethra and going to the external environment or into the female reproductive tract. All right, so this is a review from Anatomy and Physiology 1. Actually, this is a slide from Anatomy and Physiology 1. Now, in our discussion of physiology, I am going to assume that you are well versed in the anatomy of both male and female. So if you need to review this, I suggest going back to your A&P 1 notes or going back to the textbook for a brief review. So we'll kind of switch gears a little bit and talk about hormonal control. So what hormones are coming from the male reproductive tract and how are they controlled? Well, remember back to when we were talking about hormonal control in general. There were 2 main glands that we really focused on, one being the hypothalamus and one being the anterior pituitary. Your hypothalamus is considered to be the master gland. It's kind of in charge of everything. It drives stimulation of the anterior pituitary, and then the anterior pituitary is going to drive stimulation of the testes in this case. So, and we'll see a chart I have in a little bit. There is negative feedback as well. But the reason all of this is important is because the male body needs masculine hormones, specifically testosterone, to maintain, or rather start or initiate and maintain sperm cell production. And on top of that, male secondary sex characteristics, which are special features that only a male will display. And a woman will have secondary sex characteristics that only she will display that are driven by her sex hormones, and we'll talk about those later. So we really spend a lot of time focusing on testosterone, and that's probably the biggest player. But there's lots of other hormones, and some of them overlap between male and female. And I think it really would be important to do a kind of a brief review of those hormones. So the big one that comes out of the hypothalamus is GnRH, gonadotropin releasing hormone. Now, this hormone will leave the hypothalamus, and it's basically going to go to the pituitary to stimulate the release of gonadotropins. So there are specialized hormones, luteinizing hormone, or I'll typically just call it LH, and follicle stimulating hormone, FSH, are going to be stimulated to be released from the anterior pituitary by gonadotropin releasing hormone, or GnRH. Now, you're going to notice that GnRH, LH, and FSH are both present in males and females. But here's the thing that you're going to have to make the note on. They have different functions depending on the gender. All right, so LH is really important for stimulating interstitial cells within the testes. So these cells are going to drive the formation of testosterone. So LH is really directly behind testosterone formation. Now, FSH, on the other hand, stimulates those sustentacular cells. Remember, or sometimes I call them the Sertoli cells. And the FSH is really going to drive the birth, if you will, the maturation and the proliferation of sperm within the testes as is a response overall to testosterone. So it's a pretty complicated process. And to add an even greater layer of complication, this is all negatively feedback controlled. All right, I'm going to jump ahead just a slide, and then we'll come back. Well, a couple slides. All right, but I'll go back. Don't worry. I want to talk about that negative feedback really quick. So here, if you follow my mouse, here's your hypothalamus, and it's releasing GnRH. The GnRH is going to go to the anterior pituitary. The anterior pituitary is going to release FSH and LH directly into the bloodstream. And FSH and LH will flow all over the body until it finally reaches the testes. And they're both going to have a very positive stimulatory effect on the testes. The LH is going to be responsible for driving testosterone formation, and FSH is going to be really important in sperm cell production and maturation as a oversight from testosterone. Now, here's the other thing you have to think about. Testosterone is going to come out of the testes along with some other androgens that we don't focus on. And then when testosterone is created, another hormone called inhibin, which does exactly what you think it does, it inhibits, are going to be released at the same time from the testes. They are going to go into the bloodstream, and they are both, inhibin and testosterone, going to feed back at the anterior pituitary, and both feedback at the level of the hypothalamus to decrease the GnRH that is stimulating the anterior pituitary. So it's a very tight control. Now, as a side note, we have our FSH and our LH in the bloodstream, and they are also really important for stimulation of muscle and hair growth and libido, vocal cords, see here, and their stimulation of testosterone is going to be really important for bone maintenance as well. All right, so now we'll go back. Male sex hormones are called androgens. So testosterone is an androgen. There are other androgens that are present, like dihydrotestosterone. It's very similar to testosterone. It's produced in a smaller quantity, but it's a lot more potent. But we're really just going to focus on testosterone because it's really truly the most important androgen. And interestingly, testosterone secretion is really important in fetal development. So once an embryo reaches the fetal stage and it is male, testosterone secretion is really going to increase. And then testosterone becomes quiescent in young childhood. What basically means is there's not a whole lot of it. It's very quiet. And then again, when the child reaches puberty, we see testosterone start to really ramp up in quantity. All right, so this is a little bit of what I just talked about. Before birth, again, you have lots of testosterone that's driving the masculinization of the fetus. And like I said, it's pretty quiescent during childhood. And then at puberty is where all of the secondary sex characteristics come out. And these are some of the ones that I already alluded to when I was referring to the diagram on the next slide. The vocal cords getting bigger and thickening. This is where a boy's voice they say, cracks, and it becomes very, very deep. Men typically have thicker skin than women, and that's driven by testosterone. Muscle mass bulks up significantly. And this also, testosterone is the reason why men have less of a risk of osteoporosis than women. All right, so I already talked about the slides. We're going to move forward into females. So now we're going to talk about oogenesis. Oogenesis is a lot like spermatogenesis, except now we're creating an egg instead of sperm. And instead of getting, in the spermatogenesis, we got 4 sperm out of the deal. We're only going to get 1 egg and 3 polar bodies, but I'll talk about that in a moment. So starting at puberty, some primary oocytes continue on with meiosis 1. So until this point, you really just have precursor or progenitor cells that are just kind of hanging out in stasis. When a young girl is in her childhood years, her reproductive organs, meaning her ovaries, are just kind of in a stasis-like state. So at puberty, when some primary oocytes are continuing in meiosis 1, they're going to divide. And then the cytoplasm is going to be distributed unevenly. You'll essentially get 1 secondary oocyte and 1 polar body. The polar body is a lot smaller than the secondary oocyte, and it's eventually just going to die via apoptosis. Then the secondary oocyte, this is really important because the secondary oocyte will eventually become the ovum or the actual egg that can be fertilized by the sperm should that process be elected. So the secondary oocyte is only going to undergo meiosis if it is fertilized. I'll say that again. The secondary oocyte is going to be kind of frozen in meiosis 1, and it's only going to undergo that second round of meiosis if it's fertilized. So if it unites with a sperm. So think about this. How many kids do you have? How many kids does your mom have? That is the number of secondary oocytes that actually continued on and underwent meiosis 2. I have 1 son. I have only had 1 secondary oocyte actually complete and go through meiosis 2. So this is upon fertilization. If there is a zygote, that is when meiosis 2 picks up. We'll talk about this a little bit more as we continue on. All right, so here's your progenitor cell, your primary oocyte. We are undergoing meiosis 1, and we're going from diploid to haploid. But notice here at this stage, we have haploid, both our secondary oocyte, and we have our polar body. Notice the polar body is a lot smaller than the secondary oocyte. All right, so now this is where the egg or the future ovum just kind of freezes. It doesn't really do anything until fertilization happens. If a sperm reaches the egg and there is unification and zygote formation, then, oh, here's a typo. You guys fix it. This should say M2. The second round of meiosis picks up. And all the polar bodies that are created essentially are degraded via apoptosis. All right. So at puberty, the anterior pituitary on a woman is going to start to secrete FSH. It'll secrete LH too, but we'll talk about that in a minute. So FSH is going to stimulate the ovaries to grow. They're going to get a little bit bigger in response to this FSH increase in abundance in the body. So every time there's a reproductive cycle or a menstrual cycle, some of these primordial follicles are going to mature into primary follicles. So primordial follicles are these like progenitor precursors that are just kind of waiting to be selected for maturation. And if they're selected for maturation, they're going to become the primary follicles that we saw here. This is going to be your primary oocyte here. All right, so the primary oocyte is going to essentially get bigger. There's also specialized cells called follicular cells, and they're going to proliferate into something called granulosa cells. So there's lots and lots of terms here. The zona pellucida, which is a glycoprotein layer, so it is a carbohydrate protein mix that's going to form between the oocyte and these granulosa cells. It's going to be a really important layer for the egg to have. And I know that seems like a lot right now, but we're going to go through all of it in detail. So let's talk about follicle maturation in the ovary. So here is the ovary. This is this is the fimbriae and the fallopian tube. And you have, these are the primordial follicles. You know, by the way, that this is the fimbriae because these are little fingers, finger-like projections that are going to grab the egg once it has been erupted from the ovary, and it's going to travel down, or rather up in this case, it's going to move up and then down through the fallopian tube. This is a suspensory ligament. This is not a fallopian tube. All right, so you have a primordial follicle is selected. So one random primordial follicle is selected. It then becomes the primary follicle. And then as it grows, notice the changes that are occurring. It's getting bigger and bigger. So it's going to become something called the preantral follicle. And basically it takes 2 to 3 months. So you're talking like 60 to 90 days for the primary follicles to reach the preantral stage. So what this tells you is you have to have several primordial follicles selected at once because they're going to mature in different stages. And then they're essentially going to ovulate at different times as well. So you kind of have to have several going at once in order to have an appropriate ovulation every about 30 days. All right, so as we move on, we had our preantral follicle and we have our mature antral follicle. So notice how much bigger it's getting. So this one little speck here that's a primordial follicle is getting bigger and bigger and bigger, and it's really taking over a good portion of the ovary. So there is a special set of events that must occur before ovulation can happen, and we will talk about those in LH Surge and other things that will happen. And in that case, what happens is the egg, this is that future ovum, the egg is going to be physically expelled or blown away from the ovary itself, and it's going to be kind of caught by these fimbriae, these finger-like projections, as it's going to then direct its movement into the fallopian tube. Now, after ovulation, so you kind of have what's left behind here, this antral follicle is going to change. So the remnants, or kind of the pieces that are left over after ovulation, are going to develop into something called the corpus luteum. That's this kind of yellow-like structure here. So if fertilization occurs, then you would have this egg is going to then undergo meiosis 2 and continue on with implantation and other actions that we'll talk about in the uterus shortly. However, if there is no fertilization, then the corpus luteum essentially degrades and is sloughed off. All right, so a few more things about follicle maturation before we dive too much deeper. So while a follicle is developing, so the ovary itself is going to have cells outside of the follicle, and it's going to be arranged into several layers. So I'm going to flip back a slide. So when I was talking about the preantral and the mature antral, and you see all of these different colors, these gradations starting to appear, that's what I'm going to start talking about now. So the innermost layer is called the theca interna, and it produces the steroids. Steroids are going to be estrogen. Estrogen is a steroid hormone. Then you're going to have a layer of tissue around this, the outer layer called the theca externa, and this is all connective tissue. So follicular cells are going to grow and divide into anywhere between 6 and 12 layers. It just depends on where you're at in the follicle, and it kind of depends on the woman herself, but there's an average of between 6 and 12 layers. There's going to be the little fluid-filled spaces among the cells, and it essentially, we talked about anywhere from 60 to 90 days back here to get your follicle formation, or rather your primordial follicle maturation. Well, if we talk about primordial follicle all the way to preantral follicle, it can take up to 150 days. It doesn't always. It sometimes can take between 60 and 90, but this is another reason why several primordial follicles are selected at once, because their rates of maturation can vary. So the 65 to 70 days of development is going to convert the preantral follicle into a fully matured antral follicle. And then on top of that, you're going to have 15 more days. This is kind of the first half of the menstrual cycle that are going to convert the antral follicle into a Graafian or a pre-ovulatory follicle. So this is essentially getting the egg ready for expulsion out of the ovary. All right, so this is just a really cool picture. This is an ovary that was removed and it was sliced really thin and it was stained so you could kind of see what this looks like. So this is the oocyte or the egg. And then here, this little thin layer here that's hard to see, it's like a blue layer. It's acting like a border. And then you have the purple beneath it. This is in reference to the maturing antral follicle. And then here you can see eggs in different places in development. So there's a primordial follicle, really teeny tiny. And this is a preantral follicle that has not yet reached the stage that this one's at. All right. So the mature antral follicle, so this is going to be something that's ready for physical expulsion off of the ovary, is about 20 millimeters. That's like 2 centimeters. The secondary oocyte is surrounded. So here, the secondary oocyte is here. This is the egg that's going to become the future ovum. It's surrounded by a specialized mass of follicular cells called the corona radiata. And here you can see it here. And this is what it actually looks like on a piece of tissue that was stained. All right, so you're going to get several primary follicles are going to start maturing at the same time. But how do you only ovulate one? Well, one of them will become the dominant follicle, and it usually takes off and responds more quickly and better, if you will, quote unquote, air quotes over here that you can't see, to the hormones and just kind of takes off as leader of the pack. Well, sometimes a woman can ovulate 2 eggs, and that would lead to fraternal twins. But we'll talk about that in a different chapter. So for now, we'll concentrate on a single embryo pregnancy. So typically, only the one dominant follicle is going to develop, and the other ones that don't win the race per se are essentially just going to degenerate. So we talked about 150 days. Now we're going to kind of up it and talk about 300 days, because now we're talking pre-antral follicle, and now we're talking fully antral follicle. So the primordial precursor follicles that are going to be selected to become eggs to essentially rupture out and leave the ovary, they take almost a full year before ovulation. They are selected about 10 months in advance. And so that's why this is a continual process. A woman has to essentially select some primordial follicles every 10 months. 10 months, and then the next 30 days, she'll select more. That'll have to go 10 more months, and so on and so forth. You get my point. All right, so ovulation. So nearing the end of the follicle's maturation, so where we've gone from preantral to antral follicle, the primary oocyte will undergo that first meiotic division, and then you'll have your secondary oocyte and the first polar body. The first polar body will just kind of hang out and wait. The first polar body will undergo another round of meiosis if the the secondary oocyte is selected, but they'll all just degrade, the polar bodies, that is. The process of ovulation is going to physically expel that secondary oocyte or the ovum off of the ovary and into the fallopian tube. Now, one note I want to make is I always call it the fallopian tube. Your book calls it a uterine tube. Potato, potato, okay, guys? Same thing, so don't get confused by that. All right, so just as we talked about the physiological process in sexual stimulation, how it's important for sperm to leave the male body, we have to talk about the physiological process in sexual stimulation that prepares the female body for the incoming sperm. So we had the exact same headings: erection, lubrication, and orgasm with respect to male physiology. The same headings can also appear in the female category as well, just different things are happening. So there is an erection that happens in women. It's the clitoral tissues. So this is in the anterior portion of the vagina, and they respond directly to sexual stimulation. The reason this is important is because when the clitoris is stimulated, nitric oxide will be released, and it's going to dilate the arteries and allow the vagina to open up a little bit wider. This is important for the penis insertion, so the sperm can be released closer to the cervix, allowing the sperm a shorter travel time to get to an awaiting secondary oocyte. So in addition to getting our erectile tissues stimulated and nitric oxide released, sexual stimulation is also going to cause several glands. There's vestibular glands surrounding the vaginal opening that are going to start to pour out copious amounts of mucus. This is important to allow lubrication for the incoming penis so it doesn't, it can be propelled further into the woman's vagina. Finally, the last step is going to be orgasm. So the clitoris will respond to sexual stimulation. And basically there are muscles that are going to react in the perineum and the uterus, and they're going to affect the uterine tubes. So the uterine tubes themselves are surrounded by circular muscles that are going to start to contract rhythmically. Remember, the uterine tubes are the same thing as the fallopian tubes. So they're going to contract rhythmically to help propel the sperm towards the egg and then propel the egg towards the sperm so they can meet, hopefully, somewhere near the halfway point. All right, so this chart was in your notes. I just added a couple points here that will be good review. So when you have sexual stimulation in men, we talked about it being parasympathetic impulses. It's the same thing in women. It doesn't suddenly switch. It is a parasympathetic driven act. And then another thing I want you guys, we just talked about this, that those vestibular glands surrounding the vaginal orifice are going to release all of that mucus to enhance lubrication. And then orgasm is more about the rhythmic contraction of muscles and the entire female reproductive tract to propel sperm and egg towards one another. All right, so just as I provided a review of all of the structures for male anatomy, here's a review of all of the structures in female anatomy. So if you think you need to kind of refresh your memory on these, please do so, because everything we've talked about just assumes that these are still familiar to you. All right, so we talked about male hormonal control. Now we're going to talk about female hormonal control, and we'll have to talk about it in a couple different ways. So maintenance of the egg is one thing, but then there's also a very complicated menstrual cycle. So just as before, the hypothalamus is the master gland. It's going to release gonadotropin releasing hormone onto the anterior pituitary, and then the anterior pituitary is going to release the gonadotropin class of hormones onto the ovaries. So the reason this is important is because you're going to maintain secondary sexual characteristics, and it's also going to directly drive formation and maturation of the eggs. And these hormones operate in a circadian rhythm of sorts, not a 24-hour circadian rhythm, but more a 28-day cycle. So let's briefly review all of the hormones. You have gonadotropin releasing hormone that we talked about in men. This comes out of the hypothalamus the same as it does in men, and it's going to release FSH and LH the same as it is in men. Now here's where we have the change up. FSH and LH are going to have different functions in the woman. FSH and LH are going to control oocyte maturation, and they're going to control formation of estrogen. Estrogen and progesterone, more specifically. So estrogens, and notice it's plural. There's actually 3 types of estrogen in the female body, and there's 1 type of progesterone. And they all come out of the ovaries. They can come out of the adrenal cortices. So the adrenal cortex can release small amounts of these. And estrogens and progesterone are all released from the placenta when a woman is pregnant. So the estrogens, all three of them, are important for enlarging the accessory reproductive organs. They're responsible for stimulating the thickening of the endometrial layer of the uterus, and they're really important for developing and maintaining secondary sexual characteristics like breast and mammary gland and ductal development, so both. They're also going to lead to an increase in adipose tissue in the breast, thighs, and butt. And a woman's skin is more vascular than a man's skin because of estrogen and progesterone. I'm sorry, estrogen directly. Progesterone a little bit, but progesterone's main functions are going to be to kind of regulate the menstrual cycle. Progesterone has a huge effect with mammary gland development that we'll talk about. And progesterone is very, very, very important for negative feedback control of the gonadotropins FSH and LH. So here's what I'm talking about. So we have our GnRH is going to come from the hypothalamus and act on the pituitary, anterior pituitary. It's going to release FSH and LH, which are going to act directly on the ovaries. You're going to have estrogens that are going to come out of the ovary, and the estrogens, along with the FSH and LH, are going to be really important in breast development, vascularization of the skin, enlargement of the reproductive organs, making the endometrial layer of the uterus bigger and thicker, and progesterone is going to feed back and regulate hypothalamic and pituitary control, as well as really drive menstrual cycle. And it will also be really, really important in gland and ductal development in the breast. All right, so let's talk specifically about the menstrual cycle. The menstrual cycle is referred to as menses. Menarche is not menses. Menarche simply is the very first reproductive cycle, or the very first menstrual period a woman will have. After that, it just becomes monthly menses. So typically menses are going to initiate around the age of 13. This age is actually dropping. It used to be closer to 14 or 15, but over the past 200 years, the number has gone younger and younger. And a woman will menstruate or continue to have these menstrual cycles into her early 50s. 52, 53 is about the average for a woman to undergo menopause, which is the cessation of the traditional menstrual cycle and no more menstrual periods. So every month, so it's on about a 28-day cycle, there are these regular reoccurring changes. So a woman will have very similar changes in hormone cycling amounts in her body on a, like I said, about a monthly rotational basis. So a woman will essentially have one mature antral follicle, will be ovulated. It's ruptured out of the ovary and into the uterine or fallopian tube space, and then the remnants or whatever's left over from that follicle on the ovary kind of close up and become something called the corpus luteum. Now, if none of this happens, if there's no fertilization, so if the egg is released but not fertilized, the egg degrades. A woman is actually only fertile for between about 16 and 24 hours. So timing is everything. So if after about that 16 to 24 hour period, the egg is not fertilized, it begins to degenerate. And then the corpus luteum will also start to degenerate, becoming something called the corpus albicans. All right, so this is a review of hormonal control of female secondary sex characteristics. So this is something to make sure you review on your own. I'll mention a couple things briefly, but we've covered a lot of this already. So remember, the anterior pituitary is going to release FSH and LH just like we saw in men, but we start to see differences in function here at step 3. FSH is going to be really important for follicle maturation, allowing those granulosa cells to form and function correctly. And LH is really going to be really, really important in estrogen production. Estrogen is important for maintenance of secondary sex characteristics, and functions surrounding the egg. And progesterone, on the other hand, is going to be really important for regulation of the entire cycle as a whole and breast development during pregnancy. So the main events of the menstrual cycle you will need to make sure you are familiar with. I have a chart that basically is what is here in writing, but it's graphically, and I like the chart a little bit better, but I think that this also shows, it's just kind of a good tabulation of what actually happens. So the anterior pituitary, again, is going to release the FSH and the LH, and I keep talking about this over and over and over again, because the FSH and the LH are going to kind of, here, I'll skip ahead to the graph. So here we have our FSH and LH right here. LH will always be a little bit more than FSH, but they stay at kind of low levels. So just notice they're there, they're being secreted, but they're kind of at low levels. And then all of a sudden, you're going to have an uptick in LH. So FSH and LH are going to do all of the things that we already talked about, but then at about the 14-day mark, LH will swing into huge abundance. It'll be secreted in copious amounts. This is called the LH surge. And when the LH surge happens, I'm going to skip ahead to the next page, or next slide rather. If you look at FSH and LH and then skip down here and look at estrogen here in this red and progesterone here in the green, you'll notice that the LH surge happens and there's also a peak of estrogen. And then about 16 to 24 hours after that, the egg is ruptured, it leaves the ovary, and it essentially is going to wait for fertilization. All right, so if the secondary oocyte is not fertilized, it's going to degrade and the menses will commence. So basically the concentration of hormones that are going to come from the corpus luteum, because the corpus luteum is going to start to secrete hormones, or it is rather the whole time. Those amounts of hormones are going to decline steadily. So when a woman is approaching her menstrual period, her hormonal levels, as you can see here, start to plummet. So this here, right, is in reference to the menstrual period beginning. So estrogen and progesterone get very, very high about a week prior to the menstrual period, and then they start to really bottom out. And once they're at that bottomed out level, the menstrual period begins. And the one thing I do want to take your attention to here is that this and this are essentially the same thing, because you have to think about this as a continual cycle. It's a circle. We're looking at it linear because we're looking at a slide on a PowerPoint, but this step feeds into this step, and then the process starts all over again. All right, so I mentioned before that menopause is the cessation or the stoppage of the menstrual cycle. So it can occur as early as a woman's late 40s, and it can continue until a woman's in her late 50s, but that's not typical. The typical age is when a woman is in her early 50s. So the ovaries are not going to produce as much estrogen and progesterone as they once did. So they really start to decline in what they can produce. They go down and down and down and down in the amount of hormone that they're pumping out. So sometimes female secondary sex characteristics start to disappear. Women can start to experience very negative side effects that are associated with menopause. But then once menopause is reached, now menopause is not the process leading up to, but menopause rather is the last menstrual cycle. After that, most of the negative symptomology should kind of go away. All right, so let's switch gears a little bit and talk about mammary glands. Mammary glands are considered accessory organs of the female reproductive system. So they are specialized glands that secrete milk following pregnancy. So they come into maturation when a woman is pregnant, but they should not be secreting milk until after parturition has occurred. That is, after the baby is born. So mammary glands are located in subcutaneous tissue, basically at the front of the body in the anterior thorax, so that's the front chest of the body within the breast region. So there is a nipple, which is the point of expulsion of the milk out of the woman's body to a suckling infant. And then there's an area of pigmented skin that basically encircles the nipple called the areola. So the structure of the mammary gland is interesting. There are anywhere from between 15 and 20 very oddly shaped glands, and those glands all feed into a very complicated ductal system. So when a girl reaches puberty, her glands start to develop, but the ducts do not. Ductal development only occurs when a woman becomes pregnant. So these glands, which are fed into this complicated ductal system, there is alveolar ducts that branch directly onto the alveolar glands, and then the alveolar ducts feed into lactiferous ducts that eventually feed to the nipple for expulsion of milk outside of the body. So this is a little bit from chapter 23, but it makes a lot of sense to include it here. So milk production and secretion. So when a woman is pregnant, she has placental estrogen and progesterone. So basically it's the same estrogen and the same progesterone that she is secreting from her ovaries, but this is now coming from the placenta because she is pregnant and she is either developing an embryo early on or a fetus later on. So, and these placental estrogens and progesterones are going to stimulate continued mammary gland development. And then prolactin is another hormone that then begins to be released by the anterior pituitary gland. So prolactin is really important for milk production, but milk expulsion out of the body is driven by a hormone called oxytocin. All right. So a couple notes on milk. The very first milk that a woman produces after a baby is born. So when a baby is born, that's called parturition. So directly after parturition, the first milk is called colostrum. And then that only lasts for a few days. And then mature milk, so the milk that the infant will essentially live off of for the rest of the nursing period, is just called mature milk, and it's then secreted and there's no more colostrum. All right, so this is a review of basically everything that we've talked about. So use this as your study slide. All right, so I think this is really, really fascinating. This is a mammary gland. This is a slice of tissue from actual woman's breast when she's not pregnant. And then this is a slice of tissue from a mammary gland from a woman who's lactating. Notice the huge difference in how it looks. So this is the glandular tissue here and here. These are the glands, and here are the glands. So look how much different those glands look when lactating versus non-lactating. It's just so wild, the huge evolution that the cells really take between this preparatory period when a woman is not pregnant to when she's fully lactating after she has had her baby. All right, so milk production and secretion. So it really is driven by suckling infants. So a baby can even cry and stimulate it, but it's mainly driven directly from suckling. So the milk is ejected by a reflex that's stimulated by oxytocin. I already mentioned that prolactin is important for the production of milk, but it does not drive the release of milk. That is from physical mechanoreceptor stimulation in the nipple, which leads to oxytocin release from the posterior pituitary, which then allows the milk to escape the body. And that is it, guys. That's all I have for you. Have a wonderful chapter 22 studying and part of chapter 23. Look for the next chapter soon. And if you have any questions, please always email me.